Is your dad still okay? Or is
It's getting very... He's now...
Let's go.
All right, so we'll make a start. So Len Harrison couldn't make it. So today we have Associate Professor John Wentworth talking to us about type 1 diabetes, which is another one of the diseases that we're going over that overlaps into multiple different topics we've touched on this year. So autoimmunity, and also there'll be a bit about microbiota and things that Sammy talked a bit about on Monday. And so John's an endocrinologist over at the Royal Melbourne Hospital and has been working on various aspects of things that may predispose people to diabetes, and they're trying to work out how to stop people getting it and things, which of course is very topical and causing a big health burden these days.
Great, thanks very much. And look, I hope we're going to leave some time for questions. If you've got a burning question, stick your hand up and we'll deal with it on the spot. I'll try and race through a few of the background slides. We'll see how we go. I'm hoping we've got time at the end for some talk. So type 1 diabetes is a relatively rare disease, but it carries a substantial burden. And some people, particularly the paediatricians, say that the impact of a diagnosis of type 1 diabetes in kids is comparable to being told the kid has cancer in terms of the stress and also the sort of the long-term health and mental health impacts of that. Its main issue is that you've got to give insulin. You've got to learn to treat yourself with insulin to make sure your blood sugar doesn't go too high or too low. If it goes too high, you get things like kidney failure and blindness from the effects, the toxic effects of high sugars. If it goes too low, you black out and you crash your car, or you drown, or you have a shit day the next day because you didn't sleep well. And it really and carries a major burden, both in terms of the amount of effort people have to make to control the sugar levels with proper insulin dosing, but also certain limitations that it places on life. And you can see there that the economic cost just in terms of health dollars is also not insignificant. So it's a serious disease that affects several, but certainly not the majority of Australians. And it's fundamentally an immune disease. We treat it as a metabolic disease. We focus on glucose and get those levels normal. But fundamentally, it's a disease of the immune system where the immune, for reasons that we really don't understand, specifically attacks the insulin-producing beta cell within the islet of the pancreas. Okay, so the islets contain also alpha cells that make glucagon and delta cells that make somatostatin. But about half of the islet consists of beta cells. They make insulin, and the immune system goes specifically after those cells and knocks them off and moves on. So if you look at an autopsy specimen from a type 1 pancreas, you'll see there's no insulin staining, but there's still plenty of alpha and delta cells there. So it's a really specific, targeted immune attack. We've been spending the best part of 4 decades trying to work out how to turn that off safely. Here's a cartoon sort of trying to outline the key events in this autoimmune process. What's quite apparent is that it's directed at the beta cell. So we really do believe that there's some sort of loss of tolerance to beta cell specific antigens, of which insulin is the prime candidate, because very few cells in the body other than the beta cell actually make insulin, and the only other cells are circulating monocytes, and they do it at very, very low levels. The antigen is either picked up within the islet or more likely is taken either by antigen-presenting cells, or itself just diffuses off through the lymph to the pancreatic lymph node. And this is where the CD4 helper cells ramp up a specific immune attack. This class 2 MHC is absolutely critical, and you'll see from the genetics it's a major factor in type 1 diabetes risk and pathogenesis. And then the response leads to antibody production, which we'll talk about in a moment, but also primes the CD8 T cells, and through their effector arms they come back in and they knock off beta cells that are expressing these antigens through MHC class 1. So I guess you've seen this sort of diagram in the context of other autoimmune diseases like rheumatoid arthritis, but this is how we understand it to be happening in the pancreas. And it makes it very difficult to study in humans because you need to actually fish out the pancreas or the pancreatic lymph node to get any real understanding as to what's going on. And most of the time we're restricted to blood and studying what's going on in the blood in the hope that we can catch that minute population of cells that might be autoreactive. Now this is a classic scheme and figure that's been around for 3 decades in various forms and dreamt up by a guy called George Eisenbarth in the US, who is the father of beta cell islet autoantibodies. And it makes a number of very important points about type 1 diabetes. And again, it makes the point that this is primarily an immune disease, not a metabolic disease. And we really think about it in terms of how our beta cells are functioning, because at the end of the day, you don't get diabetes if your beta cells are up to the job and can make enough insulin for you. Diabetes, both type 1 and type 2 diabetes for that matter, are diseases of insulin deficiency, either absolute in the case of type 1 or relative in the case of type 2. So, you've got plenty of beta cell mass as a developing infant and young child. Often there's genetic predisposition which is dominated by these HLA class 2 genes. And then we think, and we've got good evidence for, I'll show you in a moment, that there's an environmental stress or stresses that trigger autoimmunity, so they trigger the loss of tolerance and the T cell mediated process. And it is reflected by circulating autoantibodies that we can measure in the serum. So we can measure antibodies against insulin, against GAD65, against islet antigen or insulinoma antigen 2, and against zinc transporter 8 in the blood many years before people get type 1 diabetes. And in fact, it can be a decade or more before people progress from having antibodies to getting type 1 diabetes. And so that tells you that the immune process is either very slow burning in many people, or it's waxing and waning, or that there's other modifiers of it. But it gets going early, often in the first decade of life. And then for reasons we don't understand, there's progressive ramping up of that response, loss of beta cell function. Initially, the glucose levels are borderline, and then they're frankly diabetic, where the levels go high, people have thirst, they've got polyuria because the high glucose levels cause an osmotic diuresis through the kidneys, and they get ketoacidosis as well, very sick, and we need to treat them with insulin. And this is usually where people show up to the clinician with frank diabetes, needing insulin, and then thereafter they're treated as having a metabolic problem. But there's a bunch of work going on, and I'll explain a bit of that as we go along, where we're identifying family members, because families are enriched for risk of type 1 diabetes through the genetic predisposition. We're screening for antibodies, and we're taking the high-risk people and either studying them closely to work out why things progress, or enrolling them in immune intervention trials to see if we can delay them getting type 1 diabetes. And I'll show you some of the recent data out of that, which I think is very exciting. This slide has been updated subsequently, but it makes a very important point that multiple antibodies in the serum. Remember I said there were actually 4 specificities we looked at. Zinc transporter 8 is also one of them here. But if you've got more than one antibody, your lifetime risk of diabetes approaches 80%. Okay, and in fact, if you've got 2 or more antibodies, the lifetime risk as it stands is about 85%. And this is in populations predominantly either with a high genetic risk or a family member with type 1 diabetes. And we believe it also applies to the general population, but we haven't tested that more fully. But essentially, the message here is that 2 or more antibodies equals type 1 diabetes. And it really justifies an effort both to understand what the hell's going on, but also to start thinking about what we can do safely in this context to delay or prevent disease progression. Now this slide was thrown in by Len simply to make the point that you've got to be a bit careful with your tests when you go out to screen the entire community. Now we go to family members where we know the risk of disease is maybe 1 in 30, 1 in 40 family members will get type 1 diabetes. But if we go to the general population where it's only sort of 3 in 1,000, then you're probably going to find more false positives than false negatives. That's what he's saying here. And so you need to be a bit careful in terms of which population you want to screen. And this is sort of Bayes' theorem about pre-test and post-test probability, which hopefully you've heard something about. Don't need to worry too much about that slide. This slide is incredibly important though. This is genome-wide association study data, progressively collected over the years. So the colour code here just reflects the era that these genes were identified. The blue is, well, HLA was the 70s, The insulin VNTR I'll speak about was in the 90s, and then this is the early noughties in green, this is the mid-noughties, and then the late noughties here. So really, you know, we knew most about the genetics early, and not a lot we haven't really gained a great deal of information despite the incredible effort that's gone into these genome-wide association studies. So there you get masses of people with type 1 diabetes and masses of people without type 1 diabetes, and you do SNP chips and look at the whole genome and you work out what areas of the genome are most tightly associated with type 1 diabetes. You get what's called an odds ratio or a LOD score here. It's a logarithmic odds ratio. And then you work out what the effect size of these genes is, and you can see here that the standout gene is HLA class 2, okay? And the second gene is the promoter area of the insulin gene, which affects, we believe it affects gene transcription and translation, particularly in the developing thymus, so that if you have low expression of insulin in the thymus, you're more likely to have T cells that escape negative selection and end up as autoreactive T cells in the periphery. And so this locus is also telling us, I think, that not only is it an immune disease, but insulin is one of the key antigens here that at least is kicking the process off. These other antigens, GAD and zinc transporter and IA2, are probably, they turn up later as a result of epitope spreading. So you have an initial attack on the beta cell that targets insulin, there's some beta cell death, and then following on from that, all these other antigens are released, and then the epitopes spread and the immune response broadens. Now, the other thing to say is a lot of these other genes are immune genes of various ilk. So CTLA4 is an immune modulatory protein. It's involved in T cell regulation. PN22 is involved in T cell receptor signaling. You can see interleukin 10 here, but also interleukin 2 here and the interleukin 2 receptor here. And interleukin 2 is very important in terms of not only regulatory T cell form and function, but also effector T cell survival. So again, the genes tell us that this is primarily an immune disease, and yet we treat it as a metabolic disease. And so we really, I think if we want to make a true impact on this disease, we need to think from an immunological point of view. This scheme is very interesting and makes the point that at the extremes of longitude, of latitude, you get more type 1 diabetes. And we don't quite understand that. We suspect maybe it's to do with more frequent childhood viral infections or perhaps some other environmental modifier. Vitamin D deficiency has been very topical as well. You can see here that there's a bit of purple, which represents high risk here in the middle. This is Sudan, and Sudanese people have a slightly different form of diabetes called ketosis-prone diabetes that really probably isn't the same as type 1 diabetes that I'm talking about. And here are the Saudi Arabians. And again, there's not as much of an HLA predisposition there, and you do wonder quite whether it's the same disease. But basically, at the extremes of latitude, you get extremes of incidence of type 1 diabetes. And the record holders are Finland, and we'll learn a bit more about that as the talk goes on. This just sort of makes the point that if you've got a family member, and particularly multiple family members with type 1 diabetes, your risk increases. Whereas if you're just from the general population, your risk is 1 in 400, and then if you've got a protective class 1 HLA allele, we don't quite know how it works, but if you have a certain HLA class 1, then your risk goes down to a very, very low number, 1 in 10,000 or less. Now, what's very interesting is that if you have a mother with type 1, you're less likely to get type 1 as opposed to having a father with type 1. And we don't understand why that would be either, but there are theories that involve either the protective effect of breast milk and insulin being expressed in the breast milk, or possibly the fact that the mother with type 1 is already injecting herself with insulin, and that helps the developing fetus learn about it and get a bit of tolerance to it. I don't know. Anyway, you can have a look at that slide. It's why when we look for people with autoantibodies for prevention studies, we go to families, because it just increases our pretest probability and means we have to screen fewer people to find people who are high risk and willing to enter studies. Now, this is also an important slide based on Australian data, making the point that the incidence of type 1 diabetes since records began and insulin became available in the sort of the late 1920s, 1930s, has been pretty flat up to about the 60s and then it's taken off. And this, I don't think, is an ascertainment issue. This is a real effect. We're seeing it in many countries in the world. And it can only reflect the environment, because our genes as a population haven't changed that dramatically in this period of time. So there's something about the environment that is driving type 1 diabetes, and this curve parallels the curve you would see in type 2 diabetes. Obviously the numbers would be much, much higher, but it's a similar sort of trajectory. And so you might start wondering, are there some aspects that are sort of common to both diseases? And the obvious one is the modern lifestyle and lots of food and not a lot of exercise and those sorts of things. Here's some data from Len Harrison's lab making the point that the genetic profile has changed over the decades. So here we have, well, we haven't got the numbers here, do we? So this is the 2000s, this is the 1990s, this is the 1980s, and this is the 1970s. And this is just a DNA repository of people with type 1 diabetes diagnosed in these decades. And these populations have just been typed for the high risk HLA, or the slightly high risk, and then the moderate and the low risk HLA. And you can see that the pattern has shifted over time. But what has not changed is that the absolute number of cases accounted for by these high-risk alleles is the same. So even though the incidence here was lower, and roughly, of these 7 cases, 5 or 6 of them were due to these were the high-risk genotypes. In the same decades, again, the incidence for the high risk was 5 or 6, 5 or 6, 5 or 6. So what we're seeing here is that the environment seems to be unleashing the potential for these lower risk HLA genes to cause type 1 diabetes. And again, this is sort of, indirect evidence that the environment is driving the problem, at least in part. And here's a list of potential factors, many of which are modifiable. And you can have a look through that and think about it and maybe add some of your own. We really don't know, but there's massive interest in identifying these, because obviously from a public health perspective it's very important, particularly if we can do something that's easy and safe and effective. So I said viruses are potential triggers of type 1 diabetes, either the initial immune process or, I guess ramping it up after the autoantibodies have developed. But we don't have firm evidence. Probably the most intriguing evidence we have is for enteroviruses and the Coxsackie viruses, where on autopsy specimens people have shown virus protein within the islets and also the exocrine pancreatic parenchyma. I guess, there's also a few other circumstantial bits of information that do make the case that this is a viral illness. What's quite striking about type 1 diabetes and the islets is that class 1 HLA is upregulated on the beta cell and not on other cells of the islet or the pancreatic parenchyma. And we don't really know what's causing that upregulation of class 1 HLA. Is it a viral infection of the pancreatic beta cell? Is it an inflammatory response within or nearby the cell that's causing it to upregulate its class 1? We don't know. And of course, having CD8 T cells being absolutely fundamental to the pathogenesis of type 1, again, raises the possibility that they're going in after virus proteins at some point in time. But we don't really have much more evidence than that. And again, it's a topic of further study and review. What is really interesting is rotavirus vaccine has come in the last decade, and we are soon going to have the data to be able to look as to whether that may have had an impact on the incidence of type 1 diabetes. And certainly what we have seen, interestingly, in Australia is that rising incidence has plateaued out over the last few years. So possibly rotavirus vaccine has played a part in that. Vitamin D, lots and lots of association and circumstantial evidence that vitamin D is an important factor in determining type 1 diabetes risk. Again, with the latitude differences that I showed you, less vitamin D in the northern and far southern hemispheres would make sense because there's less sunlight. And certainly lots of studies have shown that, populations who supplement infant feeds with vitamin D or vitamin D levels being low in some mothers and not in others associated with higher rates of type 1 diabetes, again, make a bit of a case that this could be relevant. But we haven't done the formal intervention to check that. And in fact, given the safety and ease and simplicity of giving vitamin D, it's probably never going to happen that you'll be able to do a proper randomised trial to test whether vitamin D is indeed an important factor or not. Okay, so here's what's happened over the decades. And you can see here that this is what's happened to our body habitus as we've started living the modern lifestyle. And what we know is that with obesity comes inflammation, and it's mainly an innate immune system inflammation centred on the macrophage, which seems to turn up and establish an immune response to expanded and stressed out adipocytes that are probably dead or dying. And so maybe their prime function is simply just to mop up the garbage. A macrophage literally means big eater, and the prime role of the macrophage in the body is to just get rid of dead tissue or foreign objects or other things, to sort of degrade them and get them out of the way so that you can remodel the tissue and regenerate lost parts. But certainly they're found in adipose tissue and in other sites as well, and even around the pancreas and within the pancreatic fat. And we've done some profiling to show that these are pro-inflammatory macrophages. Here's a flow plot of immune cells from adipose tissue. We've split them into the various populations here. And when we look at the macrophages, here's the monocytes here, but these are macrophages here. And in obesity, you see this population of pro-inflammatory macrophages with the CD11C marker, which is also expressed by dendritic cells. And in lean people, you don't really see this at all. These pro-inflammatory cells seem to be laden with lipid, which might be in part causing them to be activated. And certainly there's a theory that says, well, look, this ramping up of inflammation and perhaps increasing one's ability to process and present antigen might actually have a knock-on effect to adaptive immune processes and autoimmunity, as in type 1 diabetes. And there are snippets of data that would support that. I guess some of the strongest comes again from Len's lab from more than a decade ago, where they showed that a marker of insulin resistance here was a predictor of whether someone with antibodies went to diabetes or did not. And you can see the differences here are quite stark. And this is a metabolic measure of insulin resistance here. And insulin resistance, as we know, goes with adiposity. And indeed, there was an association with adiposity in that study as well. So this is sort of a schema as to how we might think the modern environment is perhaps feeding into both type 2 and type 1 diabetes. Obviously, when you put on weight, you do get insulin resistant. That's primarily because your liver gets stuffed with fat and doesn't respond to insulin as well as it normally does. But as I was saying, perhaps the inflammatory component, the innate inflammatory component and immune activation leads to improved ability to process and present antigens and stimulate an adaptive immune response. The beta cell failure that you see in type 2 diabetes, which is a relative failure of insulin release, is not an absolute failure, is probably coming from some sort of metabolic stress, whereas type 1 might have a component of this as well in the modern age. We don't know for sure, but there's a real possibility that it does, but is mostly driven by that adaptive immune-specific attack on the beta cell. And you can have a read of that at your leisure. Now, a couple of comments about the NOD mouse, or the non-obese diabetic mouse, which has really been the workhorse from a laboratory point of view in terms of understanding the pathogenesis of type 1 diabetes. It's really, it's really an N=1 experiment in many respects, because it's really, it's a very genetically homogeneous population. But it does nonetheless allow you to make some inferences, particularly about therapies that might work and environmental factors that might be relevant. What's interesting, we don't see in humans, is that the female mice are more susceptible to type 1 diabetes than males, and we don't really understand why that would be the case. But what is also really interesting is that the conditions you keep these mice in are absolutely fundamentally important in terms of whether or not they'll get type 1 diabetes. And you can see that if you keep them in the wild, the incidence is very low. If you keep the bugs away from them in a specific pathogen-free environment, the incidence of diabetes goes up. And then if you keep the bugs completely away from them, they'll all get type 1 diabetes. And so it's really making the point that the environment, at least mediated through the gut microbiota, is an important modifier of type 1 diabetes risk. And this point is illustrated in a different way in this slide. So a hydrolyzed casein diet, so this is sort of breaking down the protein. There still is a school of thought that believes certainly some infant feeding formulas and bovine milk products, if they've complicated high molecular weight proteins, they're more likely to trigger an autoimmune response. And so there have been studies done where they've broken down that milk using smaller fractions and casein-based diets to see whether it can indeed prevent type 1 diabetes in humans, and those data are not particularly compelling, although they're showing promise. But in the mouse, if you use a casein-based diet, you get very little diabetes. Yet when you put these mice in a germ-free environment, you get very high levels of diabetes. And these are unpublished data from Len, but they make the point again that the protective effect of diet seems to be modulated by the microbiome as well. So getting back to humans and this fascinating sort of observation that we've made, and these are the world record holders. Finland have the highest rate of type 1 diabetes, 55 per 100,000 population incidence per year. Whereas a genetically identical population that really was part of Finland until the Finnish and Russian wars in the 40s and 50s, in Karelia has a rate of 7.4 per 100,000 population, one of the lowest in the world. And so the only thing that can explain this is environment, and obviously one of the really obvious ones is poverty, because the GDP per capita is far lower in Karelia versus Finland, and going with that, one suspects is a dirtier living environment in many respects. In that it's certainly that's generally what you find in second and third world countries. Well, they've done studies on these populations. I mean, there've been a lot of studies looking at the environmental differences here and what might account for these differences in rates, given that the genes are very similar. And a very interesting recent observation was that the diversity of the microbiome seems to be lower in those who progress to type 1 diabetes. And these are predominantly Finnish kids, accounting for this blue line here, versus the controls from Karelia and also from Finland as well. And you can see here that here's where people are picking up their antibodies and signalling that they've got type 1 diabetes and at risk of progression. And the ones that progress seem to get less diversity of their microbiota. And so that means fewer types of bacteria in their stool and a less broad distribution of those different bugs. So there'll be a predominance of one species over the others. And so this isn't obviously proof, but it's certainly, again, tantalising evidence to suggest that the microbiome is playing a role. And it's important to try and delve into that in full detail, because if you give a probiotic that is effective, it's potentially a very safe and effective intervention. And I'm thinking here of your Inner Health Plus that you get from the chemist, or your lactobacillus enriched biota from Nestle or other companies. And again, some studies are underway trying to test that. In Germany and Finland, there's very encouraging association data saying that if you're in a family where you're, as an infant, exposed to these lactobacillus probiotics at an early age, before 6 months, that your risk of type 1 diabetes is far lower. And it'll be interesting to see what happens here. So as I said, we treat type 1 diabetes with insulin. Used to be done where you drew it up in a pen, but now the pen comes pre-filled. This here is an insulin pump, and that's a glucose sensor, and we're getting to the stage where we can get the sensor talking to the pump. And so the technology is sort of improving to make life a little bit easier and to guard against the high and the low sugars that have been such a problem for people with type 1 diabetes. But it's still a burden, and we've still got a long way to go. And you ask anyone with type 1 diabetes, and they'll say, well, look, if you can cure me tomorrow, sign me up. and gee, if I never had to have this disease, I wouldn't be sad about that at all. So prevention is certainly right up there on something that we've been really keen to try and work on and advance. So these are some sort of very basic things that we need to think about when we're trying to set up prevention. We firstly need to have a good test that we know is reliable in terms of telling us who's at risk and what their risk is. And as I said, we're not yet at the stage where we know for sure that the population, that we can take our antibody tests to the population and have sufficient accuracy and fidelity to be able to give good information to people from the general population. We're working on that, and we're not quite there. We need to understand the mechanisms, and as I say, there's a lot of unknowns between having antibodies and having high sugars and very low beta cell function. There's still a lot to be learnt from that, but we're working on that. And then, of course, you need to have a therapy that is safe, because people who have preclinical type 1 diabetes or antibodies alone are otherwise fit and well. So you don't want to be proposing Herculean treatments, and I'll show you a couple of examples of that, because you're going to be causing more trouble than it's worth. And again, this feeds into all sorts of other clinical problems we have. I guess one that springs to mind is prostate cancer, where we have a test called a PSA, a prostate-specific antigen test, where you can identify people with prostate cancer. The problem is most men with prostate cancer will die with it, not of it. And in fact, the evidence has emerged that PSA test has caused more harm than good in the general population, because it's led to more intervention, more complications, more deaths from surgery. And so we're rethinking how we go about using that as a screening test, and similar conversations are having in all sorts of other areas, including breast cancer screening, and obviously it's something one will have to consider very carefully with type 1 diabetes. Anyway, here's a few slides telling you what you might want to consider. So if you really want to do true primary prevention, you want to get in before the antibodies develop. This is the window that you will use. This is the window that you'll be targeting, and you'll be wanting to use something that you'll be able to roll out in a population level that is completely safe. If you're looking at doing secondary prevention where you see the antibodies are there and you want to either stop diabetes developing, stop hyperglycemia developing, or at least delay its development, then again, you want something that's safe, and you want something that works. And I'm going to show you some data from some of these interventions in a moment. And then, of course, when someone's diagnosed, you've got a bit more therapeutic freedom, because you've got someone who's got an illness. They're very keen for you to do something about it, and there's justification to hit them hard with more heavy immune modulatory therapies. There's a whole bunch of things that we've done to people over here over the time. Some have shown success, and I'll show you some of them at the moment, but none of them are sufficiently effective to justify routine use. And I think we're at the stage in type 1 diabetes, at least at this recent onset window here, that we're starting to understand which therapies are at least effective. And like chemotherapy 50 years ago, we're going to start putting them together to see if we can get synergies that really make a meaningful impact. And we haven't quite got there yet, but we're about to be, the first combination therapy trial will be reported later this year, and we're going to be stepping up some more in the years to come. I think Len chucked this slide in simply to say there's a bunch of really effective therapies for rheumatoid arthritis. And there are parallels between rheumatoid and type 1 that would suggest that we need to think about using a few of these in type 1 diabetes. And I think the other point to make here is that we don't hesitate reaching for these somewhat heavy-handed therapies in rheumatoid arthritis, because you can see the autoimmune process, and obviously there's pain and other problems with the joint that justify treatment. Whereas in type 1 diabetes, you don't see that inflammation. If you could see the pancreas, maybe you would. But it means that clinicians are very less, and patients are much less likely to embrace these somewhat hazardous or times hazardous immune therapies, because they otherwise feel well and they don't see what all the fuss is about. So here's some things that we've been trying in type 1 diabetes. The anti-CD3 molecule I'll show you some data for. Anti-CD25 was being used as a means of trying to knock down effector T cells, and again was ineffective and perhaps even accelerated type 1 diabetes. Rituximab targets B cells, B lymphocytes, through anti-CD20, and it has shown effectiveness. It was one of the first trials to demonstrate effectiveness in recent onset type 1 diabetes, and very interesting too, because you don't see the B lymphocyte appearing in those cartoons that I showed you. And certainly we don't believe the antibodies themselves are pathogenic in type 1 diabetes. And we believe the beneficial effect of rituximab is mediated by its effect to dampen antigen presentation by the B lymphocytes to the CD4 T cells. And that's probably the mechanism of action. And again, it's a sufficiently impressive effect to justify combination trials that are being planned. Blocking innate cytokines, particularly IL-1, has not been effective. Interestingly, anakinra, which is a decoy inhibitor, or it's an IL-1 receptor linked up to an IgG molecule from memory. Anakinra is anti-IL-1, and it actually has small effects in type 2 diabetes to improve pancreatic function, but in the type 1 context hasn't been effective in terms of preserving beta cell function. And more recently, I'll show you some data on abatacept. This is a molecule that enhances immune tolerance. So CTLA-4 is part of the immune synapse and is involved in I'm just trying to think of the word. Maybe you've got the word. Yeah, so it's co-stimulation. Co-stimulation is involved. Once the T cell receptor synapse is formed with HLA class 2, you need a second signal, and CTLA-4 with CD80 or 86 can provide that signal to activate the T cell. And abatacept mucks up that process and favours lack of activation and immune tolerance, and shows some effect in type 1 diabetes with remarkably few side effects, which I think is really important. Okay, I don't think we need to really dwell on that slide, but I will spend a bit of time on how we might choose the antigen for antigen-specific therapy. So here we're thinking about primary or secondary prevention, where people don't have hyperglycemia, they have sufficient beta cell reserve, and we're trying to modulate that immune response in an antigen-specific way so that we can specifically turn it off. And really, as I said, insulin seems to be the standout molecule. But we must remember that insulin comes from the pro-insulin molecule. So it's actually made, there's a few extra residues tacked on this end as well as a pre-pro-insulin. But pro-insulin is made, and then these yellow bits here are cleaved off, so that insulin comprises two chains, an A chain and a B chain, linked by two disulfide bonds. It's an incredibly stable molecule, but it needs to have that configuration to have its metabolic effect. But people have wondered, and there is evidence to suggest that these residues here in the C-peptide also play a role in the immunity that underlies type 1 diabetes. And certainly it would still fit in with that insulin gene VNTR effect as well, because obviously in the thymus this whole thing is being expressed and exposed to the developing immune system. So there's lots of good evidence that this is the one to go after. I guess the other one that I haven't dealt with is the fact that if you study children at high risk from birth through to age 10, if they get antibodies, the first antibody is generally against insulin, and then the other antibodies follow as this sort of epitope spreading phenomenon that I mentioned unfolds. So again, that's other evidence to suggest that insulin is the prime driving antigen. So how do we give insulin antigen to induce immune tolerance? And trials have been attempted, and certainly some people thought you give it under the skin, as you do when you're doing desensitization therapy against house dust mite or against your cat or various other things. But they've done that trial and it doesn't work. It doesn't make it worse, but it doesn't work. It doesn't prevent type 1 diabetes in people who have antibodies. However, if you give an antigen through the mucosal system, through a mucosal surface, either up the nose or through the gut, where there's an enriched population of regulatory T cells, there does seem to be some prospect of benefit and some prospect of tweaking that immune attack away from insulin into a more regulatory state. And there's a bunch of mouse data that will support that, as is listed here. And Len Harrison has shown quite convincingly that actually giving it up the nose seems to be more effective than ingesting a large amount of insulin and having it survive degradation in the stomach to get to the small intestine immune system to cause a regulatory response. Just on that issue of the mucosal immune system, it sort of makes sense that it is an anti-inflammatory system because there's all sorts of antigens, bacterial and food antigens that are coming through our guts that we don't want our immune system to respond to because they're never going to be a threat to us. And so one of the key roles of the gut immune system is to really temper the immune system's enthusiasm to go after new antigens. And so it seems giving an antigen through this system improves your ability to set up a tolerogenic response. Here are some data here from a human study where oral insulin was used in kids who had 2 antibodies and therefore were very likely to get type 1 diabetes. And in the overall study, it didn't work. But when they did a post hoc analysis, which is always, considered not definitive, and they just focused on the kids who had an insulin antibody when they entered the study, they saw a difference. They saw a delay of probably 2 or 3 years in progression to getting type 1 diabetes. And this was in 2005 published, but it was actually, the result came out in the early noughties. And because it was so enticing, they actually went to the effort of repeating the study in a population just with insulin antibodies. And we've just finished that study, and it didn't work. But it looked promising. But statistically, there was no difference. Again, it was going in the right direction. One of the very likely issues here was that compliance was not full. They've done a compliance analysis, and they have shown that people who took more than 85% of their tablets, if you just looked at those people, they certainly had a different trajectory from the control group. But overall, if you propose to give this therapy as a capsule a day in kids at high risk who have an insulin antibody, it doesn't seem to work. At least it didn't work in this case. However, they were smart enough to design this study with 3 different groups. So when you design a clinical trial, you sort of have to have a group and then you randomly allocate people to that group. And one of the groups they had were kids who were very close to diagnosis. They had relatively low beta cell function, and so they were more advanced along that cartoon that I showed you. And in those kids, very few of them, there were only 50-odd kids in this group, but there was a significant impact of the oral insulin. And it was somewhat counterintuitive because we didn't think that this relatively weak immune therapy would be all that much use if you didn't have much pancreatic beta cell reserve. These kids had much lower beta cell mass when they came onto the study and they got the drug. Yet it seems to be most effective in these kids. And that really asks some very interesting questions about what the immune system is doing over the course of the pathogenesis of type 1 diabetes. And to me, it suggests that as there's some ramping up of the autoimmune process that leads to diagnosis very quickly. And that if we can identify that time that process ramps up, maybe that's the time to start intervening with this or other more heavy-handed interventions. I really encourage you to just take some time to look at this website. This tells you a lot about what the prevention effort's trying to do in clinical trials. This slide makes the point that nasal insulin, so this is giving it up the nose rather than as a capsule, is the only therapy that's been shown to cause immune effects. This dose of 7.5 milligrams they chose for the oral insulin study was just made up. It was really, a balance between what they could do, what the FDA in the US would approve, and what seemed to work for mice. But it's actually, the dose is about 1/100th of the dose that they gave to mice to demonstrate that it could prevent type 1 diabetes. And they don't have an immune readout of oral insulin that shows that it modulates the immune system, whereas with nasal insulin, you can see here that it does have an effect. So this is quite a complicated scenario. These are people who were adults predisposed to getting type 1 diabetes, and at some stage after they were exposed to nasal insulin, they went on to insulin therapy. And when you start injecting insulin, you get antibodies against insulin. These adults actually didn't have insulin antibodies to start with because we don't often see them in at-risk adults. It's usually the GAD antibody that we see. Now you can see here that those adults who had the placebo got their insulin antibodies as you would expect, whereas those who'd been pre-treated with nasal insulin got lower titers, telling you, I think, that there's insulin-specific effects of the nasal insulin vaccine. And there's various other antibody-based assays and, in fact, some T-cell assays that demonstrate that nasal insulin can modulate the immune system in ways we believe are tolerogenic. And so that's why it is of interest as a therapy moving forward. Now, this trial has just been completed. You'll have to look at the website for full details. It was like the oral insulin study. It took kids who had antibodies and were at high risk. They could have had an insulin antibody or not an insulin antibody. It didn't really matter. And it's just been finished. And the initial data is it hasn't worked. But we're still going through the data, and I can't really say too much more about it because I don't know. We haven't got there yet. But again, just insulin up the nose in high-risk people doesn't seem to work, at least in the form that we gave it. This slide, I think, is really fascinating because it makes the point that you can actually make a massive impact on diabetes if you're prepared to accept quite a high deal of therapeutic risk. There are some crazy people in Brazil and in Poland and in China, kids as well as adults, who have bone marrow transplants when they get their type 1 diabetes. And this is based on some observational work both in kids getting bone marrow transplants but also in a lot of basic mouse data, that for some reason wiping out the immune system and then having it regenerate, much like turning your computer off when it crashes and having it reboot, seems to reset the immune system. And the key slide here, I think, insulin requirement going down after transplant is a pretty good sign that you've done something to preserve beta cell function. Better metabolic control by the HbA1c here and more C-peptide. I mean, it's a lay-down misère effective therapy. And you can see here, quite interestingly, out to 4 years, these shaded bars represent the people who don't need insulin to treat their diabetes. So it's an incredibly effective therapy. We have no idea how it works. And 1 person out of 80 died as a result of it. And many people had other issues, including male infertility and various other things. So it's not something we routinely embrace, but it does make a very important point that there's something you can do, and presumably it's immune-mediated, the effect. Now, there is a group arguing that actually, no, it's not actually to do with wiping out the immune system, it's about aspects of the conditioning therapy, in particularly the GCMS, the GCSF, the granulocyte colony stimulating factor, and the anti-thymocyte globulin, which is an anti-T cell globulin they give before they do transplants. And they're actually about to report on that study, the first combination study in type 1 diabetes later this year. So it'll be interesting to see how that one goes. These are some sort of recent data from some studies we've been involved in. And these studies all are about people who've been recently diagnosed with type 1 diabetes, and you give them an immune therapy and measure the effect. And you measure the effect by looking at how well they make C-peptide in response to a mixed meal. And these 3 therapies at their 12-month point here all seem to have been effective, albeit they marginally. They haven't completely abrogated the decline in beta cell function here, but they've certainly done better than the placebo groups here. And so again, we have evidence that imatinib, which is a tyrosine kinase inhibitor that has immune effects as well as metabolic effects, and we don't actually understand what the immune effects are. Teplizumab, which targets CD3 and so knocks off T lymphocytes, and abatacept that I talked about targets the CTLA4 co-stimulation pathway and enhances regulation. They all seem to have an effect, and we're going to be looking to combine these in various ways because, again, the safety looks pretty good. Okay, a couple of finishing slides on transplantation, and you've got a few minutes still. This is happening across the road at St Vincent's Hospital. What you need to do, so this is reserved for patients who have profound hypoglycemia such that they're unable to really function. You know, they're blacking out because their sugars are dropping precipitously and they can't really get their insulin right to cope with that. And it is a big problem for, fortunately, a relatively small number of patients. And these people elect to have islet transplants. Now, you can have a whole pancreas transplant, but you generally get that in the context of a kidney transplant. So if you've got renal failure and type 1 diabetes, you'll often be given both a pancreas and a kidney, and that's a fantastic solution for your problem. But islets are reserved for people who have good kidneys but still have profound hypoglycemia. They isolate the islets by basically taking a donor pancreas and chopping it up with some digestion enzymes, and then they do a lot of quality control to make sure that they're not infected and that they're looking as though they're going to be able to make insulin, and then they get infused back. And the way they get infused is demonstrated here. Under ultrasound, they identify the hepatic artery leading into the liver, and they pop a needle into it and they infuse the cells. And then they often need to do this, repeat this once or twice after a few months. They need to put the patient onto immune suppression so that they don't get rejection of the transplant, but also to dampen down the ongoing autoimmune attack. So it's pretty clear that even though you give these people have not had much of their own beta cells for many years, that the immune system can still recognise them and can still wake up and attack them. There's still immune memory. So it's got its own problems with the side effects of immune suppression, but nonetheless, this slide just shows it's a relatively painless and simple procedure. But there are problems, and the biggest problem is that there's a massive shortage of human donors. You actually need 2 or 3 pancreases to supply enough islets for one person, and obviously that's 2 or 3 pancreases you could just put straight into someone having a kidney pancreas transplant. So that's an issue, and they're trying to work around that by working up xenograft models. So there's actually a bit of a push to see if we can safely farm pigs with various immune deficiencies and modifications that will eventually permit transplantation of the islets. We've obviously got to give immune suppressing drugs. And then there's all sorts of issues with function and engraftment as well. And finally, you've got to still deal with the autoimmune issue. Look, I'll leave this slide up just to sort of tell you how we're thinking about both the pathogenesis and how we might intervene to prevent type 1 diabetes. And I guess we've got a minute or two if people have some questions that they'd like to ask. Thanks very much. Got a question?
You mentioned earlier with the treatment with insulin, you mentioned something about the inspiration of insulin administration, blood sugar monitoring. How exactly does that work?
Yeah, so the question is, how do you link glucose sensing with glucose delivery in what we call the artificial pancreas? Well, I don't know how it works, but the bioengineers can get it to work. So really what you need for it to be useful is a reliable sensor. So you need to know that the sensor is going to give you accurate data about your glucose, otherwise you're in a lot of trouble. And then, that wireless technology allows the things to talk to each other. And then the other key component is having an algorithm that actually can deal with and learn about, the individual factors that are at play here. Now, you still need to tell the system when you're going to eat. Generally, you need to do that so that it can sort of be ready to prime with some insulin to deal with that food. But they've got it working, and it's going to continue to be refined as a treatment approach. The problem with it is that it's still really burdensome for patients because it's actually more complicated than having 4 injections a day, because you've got to be attached to this thing the whole time, but you've also got to learn how to use it, and you've got to learn to deal with the error messages that come up quite frequently at the moment. So it's actually, for some people, it's too much, and I can understand. Question up here?
I was really having trouble understanding how the anti-CTLA-4 antibody.
Yep. Yeah. So why does the anti-CTLA-4 antibody protect? So CTLA-4 and CTLA-4 on T cells and CD80 or 86 on the antigen-presenting cell synapse to drive the immune response. Okay, it's part of co-stimulation, right? Yes, so there are antibodies. I mean, unless I've got this wrong. Have I got this wrong?
See, I think CTLA4.
It's an inhibitor. Okay.